So here we are looking at the slide with the 4x objective dialed into place, and I'm going to bring it into focus here. And you know you're looking at your cells when you move the slide left to right, and you see that. And you can see there's a thick area over here, and I'm going to go for sort of the edge on this smear, and I'm going to bring it in to focus and then center it right around the 50 on the ruler there. And we'll go up to the 10x objective, so 100 times total magnification, and it's already mostly in focus. So all I really need to do is turn my fine focus knob there. Again, I'm going for the edge right here because I know the cells are going to be a little bit more spread out than if I were to look over here where the cells are all really thick. So I'm going to go back out to that edge area, go up to our 40x objective, bring it into a little bit sharper focus, and then swing that 40x objective out of place so that I can come in here and put my drop of immersion oil right on my specimen. And while I'm doing this, I'm also going to come underneath and make sure that I reset my iris so that it opens up the eyes. And if you look at that, you can see there's a lot more light coming through the specimen there. And now I can swing my 40 or my 100x objective into place so that it is actually touching the oil right there. All right, so it's mostly in focus, and we'll use our fine focus knob to bring it into focus. And there you can see the results of my gram stain. This is my positive control, and you can see that if you look right around the 50 and the 60 there, that there is a little spot where it's kind of pink. That is cells that were either over decolorized, or with the gram stain, thick cell walls are food. And cells will start to eat their peptidoglycan layer, and gram-positive cells will give you what's known as a false negative. They are giving you negative results that are false because they're actually positive, but they turned out negative. And if I look around the smear here, what do you see? I see purple. The majority of the cells here are purple, and you can look around at different fields here to make sure that it agrees. And here you can see some clumps, and when you see this mixture of purple and pink and the cells are the same size and they're the same shape, more than likely you have an old culture. And this culture is more than 2 days old, but it was available, so I grabbed it. And you can see that we get that mixture of these things. Let's see. Unfortunately, this keeps focusing on the ocular there as opposed to the cells. A little bit. So if I'm looking at this, think about what is the size and the shape and the arrangement of these cells. We're using the 100x objective, where one space on the ruler is 1 micrometer in size. There is my gram-positive control. If you want to, you don't have to refocus this. You can just move it over to the side where your smear is and swing it out again, because oftentimes you're going to have to add a fresh little drop of oil on the other specimen there. So put your drop of oil right on top and swing your 100x objective back. Here I just dialed my smear back to the 10x objective so you can see it's mostly in focus. And then we'll swing it over to the 100x objective with our oil on it. And you can see it's mostly in focus, so you just need to rotate that fine focus knob a really small amount. Here is my cells with the 100x objective dialed into place, and this is my gram-negative control. And if you look closely, you can see that all of these cells that are visible in my field are that pink color. So this tells me that these cells have a thin peptidoglycan layer and an outer membrane around the outside. Now we'll repeat the process and take a look at our unknown in the middle. Here we can see the unknown bacterium that I got off of that plate there. And if you look closely at this, you can see sort of this spotted purple color. So these are gram-positive cells, but fortuitously, what we have here is these cells have actually made endospores. And if you look closely in those purple cell bodies, you can see this area where there's an absence of color inside of that cell body. And so that is telling me that these guys have made something that is resisting the gram stain. And so this is something that we would want to go on to study further with an endospore stain to really get proof that these are actually endospores, because here we're seeing an absence of color, and with the endospore you get a presence of color there. So it's a positive interaction. But we can use this to look at the size and the shape of our cells and their arrangement. And it looks from these guys that we're looking at some gram-positive bacilli. And they're probably about 3 to 4 micrometers in length. You can line them up with the ruler there and measure out the length of these cells. The reason why we see this pink cells all over the place is those are cells that are dying off here. So when they're in the process of making those endospores, they are going to start consuming their cell walls a little bit. So for a lot of these, the cells have probably already made spores and then they've died off. So there you can see the results of our gram stain. Remember to finish off our process here. Lower your stage all the way down. Swing it so that your 4x objective is dialed in there. Crank off the light and then come in and make sure that we blot the oil. You can see the oil on our lens paper there. And do that a few times. And if you want to really be nice for the next person, then you can come along and grab a little bit of lens cleaner, put a few drops on some lens paper, and hold that right up against the base of your objective lens there. And if you hold it there for a few seconds, that's going to give it a chance to clean off the objective. And then you can come in and just keep patting it down, not wiping, just blotting it to get the residual oil and Windex off of there. It's also kind of polite to clean off the 40x objective for the next person in case you got any oil on it. And if you did get oil on it because you put that blue banded objective through the oil, please clean it off immediately, because that oil is going to make it very difficult to visualize your cells with the 40x objective. And that is it for our gram stain.